Hello and welcome to this webinar titled Fully Continuous Biosimilar Manufacturing Framework, a Case Study, hosted by Biopharma Asia magazine and presented by Samir Varma, Head of Manufacturing at Enzene Biosciences, and Lotta Molander, Global Product Manager at GE Healthcare. My name is Oshreya Balog and I will be the moderator. Before we begin, I would just like to remind our viewers that there will be a live Q&A session at the end of the presentation. Audience members can send their questions at any time during the presentation via the Questions tab located at the right-hand side of the screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our first presenter, Samir Varma, Head of Manufacturing at Enzene Biosciences. Samir has more than 16 years of experience in the biotech industry, having worked at various mid-sized biotech and large pharma such as Medrex, Bristol Myers Squibb, Celgene in New Jersey, USA, and Lupin Limited and Enzene Biosciences in India. He has extensive experience in all aspects of process development and cGMP production of therapeutic biologics. Samir holds a master's in biochemical engineering from Drexel University, Philadelphia, and an MBA from New York University, New York. He currently heads the manufacturing department at Enzene Biosciences. Enzene Biosciences is a full-owned subsidiary of Alchem Laboratories and is based in Pune, India. Welcome, Samir.
Thank you so much. Thanks so much, Ursula. So again, thank you so much for giving the opportunity to kind of do this webinar. So as it says, it's a fully continuous biosimilar manufacturing framework, a case study. And so the way we want to structure this conversation is that we are right now building a plant in Enzene Biosciences, and what I would like to do is kind of share the experiences of why exactly we're doing it, how far we have reached, and what is the future of continuous. So let me take it forward. So as I said, what I'll do is I'll quickly give a quick background of Enzene Biosciences, what exactly we are, what exactly we do. So as Ursula mentioned, we are a fully owned subsidiary of Alchem Lab. Alchem Lab is about the fifth largest pharma company in India, and they are fund masters over there. There are four verticals that Enzene Biosciences is having their products in. The first and the biggest one is biosimilars. So that is our main source of future revenues. And then we also have a very good, robust synthetic peptides portfolio. And we are also working on phytopharmaceuticals and novel molecules. So these kind of four verticals. But as I said, biosimilars is the largest one. So we are working on the biologics part of it the most. And quickly, our company profile about the talent pool itself. We have scientists and engineers and kind of business leaders from different countries and different parts of the world, experiences in different parts of the world, and they've worked with different industries and different biotech companies and pharma companies. So as we can see, we have people from Amgen, Agilent, Biocon, Bristol Myers Squibb, Celgene, and you can read the rest of the stuff, right? And then we have people who have had their education from Merck. So in short, a pretty good, interested, and motivated set of people who is actually doing the work here. And what exactly we do? We have a complete set of operations here. So we have actually three sites here in Pune, Maharashtra, in India. The first one is the R&D site, which we want to call it Unit 0. That's where we do our cell line development. So we have a full-fledged group that actually does cell line development. And upstream process development, that's where we do our perfusion technology, and kind of develop the process for the biosimilars. We have a downstream lab where we do the continuous downstream. So we test out different ways to do this continuous downstream process. Then obviously we need an advanced analytical lab to test out are we doing the right thing or not. So we have a pretty extensive set of equipment for that. We also have a drug product development group that actually develops the formulation part of the drug. So those are the different departments that we have in Enzene Biosciences. And as I said, this is the R&D group. And then we have another Unit 1, that is the manufacturing area. So this one is actually This building is coming up right now as I speak, and this will be around 150,000 square feet of built-up area, 150,000, 200,000 to be honest, with the utility buildings. But this will actually have a complete, fully continuous mammalian DS process. So we are actually building this on the basis of continuous bioprocessing. And that is the most interesting part of it. And when we actually started doing it, we didn't know, we will be probably the leaders of this one. But by the look of it, it seems like, we might be probably the first one to actually fully continuous bioprocessing unit in the world itself. So that's a lot of responsibility, to be honest. And it's actually really interesting. I mean, just to build a site, scratch up, using that technology and seeing how the facility would actually look like when you have a continuous. So it will also have a drug product suite where we actually do all the fill finish part of it. But this one is actually coming up. So we wanted something in between, so we actually built a unit 2. So that is our kind of a pilot scale, a smaller scale DS production facility. So we have a fully continuous GMP manufacturing, so it's about 20,000 square feet. And the unit caters to DS manufacturing requirements for CT material. And here, as you can see in the pictures, we actually have all the equipment. So this is where we do actually 50-liter scales of continuous runs. And it has been really exciting, the knowledge and the challenges that actually brings up. So I'll talk about that a little bit more. So I just wanted to give you a good an overview of where we come from, what exactly is Enzene doing. So that's done. Let me get into the meat of the presentation. That is continuous bioprocessing. So this is something that has been really interesting, personally for me, for the past 2-3 years, for honest, and Enzene also we've been working on it for the 2 years. And the more we think about it, the more we feel it's actually going to be the most kind of disruptive technology in quite a few years in this bioprocessing industry. I mean, as you know, the bioprocessing industry is a little lagging behind in innovation. So, and probably the last big innovation that came up in this industry is the single-use technology. And people are still catching up to that. And we feel that is going to, this continuous bioprocessing is going to be this disruptive technology that is going to actually take it the next step. So my presentation would be about why do we need continuous and how we can do it, and that's going to be the meat of it. Then obviously I'll give as much details of how we are achieving it in Enzene. Some of it obviously is patent related, so I won't be able to give a lot of information on that aspects of it. But feel free to ask me the question at the end of the presentation. So continuous bioprocessing, the various strategies that we have. So continuous is it's interesting. The word, the terminology itself is different for different people, and that's what we realize when we talk to vendors, because we have heard continuous bioprocessing coming up in different places. But do we have fully continuous? So let's define what is continuous bioprocessing. What are the different strategies? So continuous upstream, the perfusion part of it, and this could be so there could be 4 different ways to do this. Right, and probably a lot of in-between stuff also. So the first one is a continuous upstream. That's called Perfusion Bioreactor with a batch downstream processing. So this has been in practice for quite some time. So perfusion technology is not new. I mean, I remember when I started working in the lab, in Medrex, we used to have a perfusion technology. We used to actually have spin filters that can do perfusion. And one of the big reasons people used to do at that time was because the productivity of the cell line was pretty low. We used to get only 0.2, 0.3 gram per liter, and the batch process would not work in a large scale. So perfusion was the best way to do it. And also the product quality, the product stability, that also was a little iffy. So the thing is, the good part was the perfusion is a good model to do it. Then people, as the cell line productivity increased to 1 gram per liter, 2 gram per liter, then we slowly moved to the fed batch part of it, and then the perfusion technology got lost a little bit. And but now with the new continuous bioprocessing, continuous the perfusion technology is coming up again. But in this case, the first way of doing it, we do continuous upstream and then with the batch downstream. So basically, the bioreactor size might be lower, but the downstream processing part of it is practically the same as the fed batch. Now the next way to do it is batch upstream and the continuous DSP. So this has been a little earlier, so it probably started with the advent of continuous downstream chromatography skid that many of the vendors have brought in, including the one that the presenter is going to give after that, so GE actually has the Reactor PCB. So those kind of technologies came, and it was possible to do continuous DSP. So this is a good way to increase the efficiency in making increase the efficiency in downstream processing. But as and it can be good for companies that already have existing technology for batch production. So already it's already in the market, like they already have the commercial license to do it. Then they will not be able to change the process. So this is a good one to actually do it if you have a batch process. The only difference would be a continuous downstream processing. So now the thing is, continuous downstream processing, when we say it's actually, we're talking about a lot of smaller micro batches. So that is another thing that we got to be a little bit when we think about continuous, sometimes continuous might not be a continuous flow of drug products. It might be actually very small micro batches coming out. So that could be an interesting concept when we are actually talking regulators also. And also the third one is actually doing having the fed batch, concentrated fed batch, and then doing using that higher productivity, higher produce in the bioreactor to do the continuous DSP, connected to continuous DSP. So that's called intensified fed batch with a continuous DSP. This is a good solution, if you want to increase the productivity in the fed batch reactor and then actually do a continuous DSP part of it. Then the final one, of course, is the fully continuous. So continuous upstream connected to continuous downstream. So this is the technology that will probably the most challenging, of course, and also the most rewarding one. So why exactly? What exactly? Why should we do fully continuous? Let me explain that in the next stages, next slides. So so that we are on the same page. What is fully continuous bioprocessing? Let me explain that a little bit more. Now, classical batch bioprocess, what is happening is that, we have wild thaw unit operation, then you have a seed expansion, then the seed is expanded N-1, N-2, N-3, depending on how many, seed bioreactors we have. Then it goes to production bioreactor, then it goes to depth filtration, whole tank, protein A, then again the product is actually held in the elute of the protein A is held in the whole tank. Then that thing is shifted to viral inactivation room where the viral inactivation happens, and then it again shifted to a Chrome 2 group, what exactly whole tank chromatography. So you can get the gist. There's a lot of whole tanks and larger equipments involved in a classical batch process. So the real estate required for this kind of a process is pretty huge. I mean, if you look at somewhere around 1,000 liter fed batch process itself would require at least 2,000 to 3,000 square feet for sure, or more than that, depending on how productive the process is. But again, if you look at fully continuous, if you're looking at the fully continuous, where upstream and downstream is connected, and we're getting a continuous stream of product. When I say stream, again, these are smaller micro batches, but we can consider it as a stream. So the thing is, now here the thing is, if you see, all the whole tanks are gone. So there is no point of holding something in a continuous process. Obviously, there could be circuit breakers, there could be a smaller surge tank, but the idea is eventually, if your process is robust enough, you should not have a surge tank. I mean, if you have enough controls in the place, you should not be having at least large surge tanks. I mean, if you can probably have smaller tanks as just a circuit breaker where we can stop the process if something bad happens in the next unit step. But again, no big large whole tanks. We are not collecting our perfusion fluid on the harvest from the bioreactor and then processing it. So we don't need collection tank for that. We are not collecting any of the protein A coming out, and we are not even having that large, huge tanks for viral inactivation. So all the whole tanks is gone. And our calculation shows what is happening is that just because of that, the amount of space required to actually do this bioprocessing drastically reduces. It goes from 2,000, 3,000, even 4,000 square feet to close to 200. That's the amount of space we require. Because everything, the scales comes down because the whole tank. And another thing that I'll come to in different stages is that the bioreactor size also decreases. So the thing is so you're basically getting a lot of savings on the real estate part of it. Again, another representation from one of the papers published, how the continuous process. So you have a 200 liter bioreactor. It's connected to ATF that is providing all the fluid that is required, and then we have protein A columns. Again, this has 4 protein A columns. So it could be either so the whole idea of continuous DSP is that you we actually process they load the first column, and then when the first column is being loaded, the rest of the three is getting washed, dilute, and kind of getting prepared for loading. So that's the whole idea. So we have different technologies. We can do a 4-column process. We can even do a 2-column process. More than that, depending on the process, depending on the yield of the bioreactor, there is multiple ways to skin the cat. So that is the interesting part of it. Then it goes to a low pH loop. Now here, this is an interesting thing that is going to happen in continuous, is that the viral inactivation step, which is relatively simple in a batch process, now is going to be probably the most complicated one, because we have to have a methodology or technology to actually make this viral inactivation continuous, holding something at a certain pH for 45 minutes and then neutralizing it and pumping it out. So there are different ways to do it. There is there are technologies to do small micro batches, or we can have a continuous flow of it. I have seen pictures of something that looks like a transformer where the entire fluid is working out. So those kind of things, technologies are there, but it's not none of the larger companies have actually come up with a very good solution of it. I mean, there are companies actually doing it, but it's actually for larger scale. But I mean, just for the scale that we actually required, we did not get it. So to be honest, we are actually building something that could serve our purpose internally. So that's how we are going to do our inactivation. And then obviously it goes to the ion exchange chromatography thing, and it goes to the. So here what we're doing right now is that we're actually holding it. So it could technically go from the Chrome 2 and Chrome 3 to directly to a filter and actually get the DS out of it. Ultracentrifuge. We have right now technology to do some part of the UFDF continuously. We have single pass CFF technology, things like that. So that could be done, but at this stage, what we're trying to do is we want to collect that in a V minus area the whole time, and then filter it into the V minus area. So the some of these steps we are slowly taking it continuous. I mean, depending on how much of experience we have in doing that, and also how much of what is the risk in taking this continuous? So what we figured out is if we make it continuous to V minus, then actually we do a V plus area, then do the final nanofiltration into a V minus area. That is probably a better way to do it. So it could change. I mean, and working here for 2 years, things have been changing. We have the R&D department is actually getting a lot of improvements in their process. So as I say, this process is a little dated. We are getting with version 2 and version 3 of the process, to be honest. So that's the fully continuous that we are talking about. And how does it work? And what are the data for that? So this is actual data from one of the runs we had. As you can see, this is the productivity increase that you get with perfusion, and it's a good cell line. So as you can see, if you see this chart, the red one is actually the perfusion, the productivity of the perfusion reactor, and also the productivity of the fed batch. Okay. So the fed batch, as you can see, it goes to I think the scale of this reactor is about 4 liters, I guess. So we're getting around 1 gram per liter, 1.5, something like that. We get around 3 or 5 gram per liter, 5 grams to the end of it. And the continuous one, it keeps producing, and that's the thing. And we can actually get around close 10 times the productivity from the same reactor. The 4 liter, we're able to get 27 grams of product. So that's a good part about perfusion. But obviously, the challenges are that, it's a continuous process where somebody actually need to tend it as a baby, or for the full 30 days. I mean, it's not feed it and let it go. So that is a catch for that. But again, but as you can see, obviously we can one kind of thought process would be that okay, we can take 2 batches during the same time, but still, even if you take 2 batches in a span of 30 days, 20 days, we are still only getting about 10 grams, whereas here, we are actually getting close to 30 grams. So still the perfusion, of course, is a much better answer to kind of increasing the productivity. And another interesting concept with perfusion is that it kind of reduces the scale. So our calculation shows that again 1 to 10 ratio. So for example, if the batch process requires 1,000 liters, I can do that in 100 liters. So the production process I showed 200 liters equivalent to actually 2,000 liters of fed batch. So that's the calculation that we can see. And as I said before, that actually reduces the footprint. So once I'm going from a 2,000 liter to a 200 liter, suddenly the square feet area is smaller. Either the amount of utilities are required to maintain that particular space is everything kind of comes smaller. So I have a slide for that in the later perfect. So and again, so this is the continuous chromatography. So this is a representative one. We have been using pretty successfully. So these are like, basically, it looks like micro batches, to be honest. So like, kind of we have these peaks, like, where the protein is eluted, and like, we kind of collect it, and that is being processed, right? So that is a quick, like, kind of science behind what we are doing, how exactly is the continuous processing that, like, that is working, and that's the kind of a little bit of science. Obviously, like, there's a lot of more information that is there, but I mean, it's at that stage where, like, we want to publish it right now. Probably, if somebody's interested, we can have a CDA and, like, kind of talk more about it. That's always a possibility. So after, like, kind of briefly explaining the science behind it, let me look at the business case. Now the thing is, like, if I can convince the people who are shelling out the money that continues is the way to go, like, I feel like, that technology can move a lot. So first thing for a business case for this is substantial reduction in capex. As I mentioned before also, it's about 5 times. The way we calculated, like, the smaller footprint, smaller equipment for process, no whole tanks, everything, it kind of comes out to be much smaller scale. So to give a quick ballpark, like, so this, like, 150,000 square feet area, like, that we are building up, most of it is taken by our DP facility, to be honest. Like, I only have around, like, 20,000 square feet. In that, I am actually having, like, 6 suites of 200 liters. And that particular facility is going to produce close to, like, 400, 500 kilos of product. So that is equivalent to, like, one annual, like, kind of production capacity of, like, of compound like denosumab. So that's the kind of amount of productivity we can get from a 200 liter from that 20,000 square feet. So that's per square feet also, like, is actually is a pretty good. It works out, like, pretty good. So again, like, there is a significant reduction in OPEX also. It's by at least by half. Most of it is actually due to the low consumption of raw materials. So this is a little bit interesting, is that, like, consumption of raw materials, like, the media might decrease, like, might not decrease that much, because the thing is the media, like, we're going to pump in a little bit more, to be honest. But then again, that is more diluted. So the thing is the overall cost of it might be actually a little bit less. But the biggest reduction would be in resin. So the thing is, once we go to, like, multiple columns, we're talking about very small columns and running it through. So we don't have any problems of storing the resin or, like, reusing it later. We use it and throw it, because these are very small columns, and we will be running, like, close to 100 cycles for each of them when we complete the batch. So that's the resin utilization is maximized. So that's one thing from the raw material aspect. Consumption of pure steam, WF electricity, all that is going to reduce drastically, because having a very small site, electricity, of course, is, like, dependent on how many HUs I have, like, what kind of temperature, pH, RH I need to maintain. So that is going to reduce a lot my like, pure steam consumption. If I use it, less whole tanks, and also, like, if I use single use technology, my pure steam for SIP part of it goes pretty low. So that is again, like, a significant reduction in OPEX. And shorter construction time. So just to give a time frame, like, it took us only about, like, 2.5 years to get, actually less than that, to get, like, from conceptual design to actually construction part of it. And, like, so we are about to, like, get it commissioned by next year, like, first quarter. So that's actually a pretty good, that's a good solution for, like, smaller companies like us that want to get a product and, like, get it up and running. And again, the capacity utilization, that's going to be 100%, because it's continuous, like, every equipment is being used continuously all the time. So again, like, just a quick slide on number of capex. I already described the entire thing. Number of vessels required is going to be reduced, hence the manufacturing area required will be much less. Again, bioreactor, much smaller bioreactors required, lower bioreactor capacity, and less number of vessels means significant reduction. So this slide is, like, I'm probably repeating myself. So I'll go to the next one. Again, as I said, like, smaller capex is lower civil costs, reduction in HVAC costs, lower paneling costs. So we are talking about much less rooms. I mean, to be honest, like, the entire bioprocess can be done in one room, because the thing is, like, the equipment is smaller, like, it has to be, like, within, like, close proximity to each other. So it's actually pretty low, and the WFS steam distribution cost is going to be small. And combining smaller bioreactor and SU technology, steam requirements considerably reduced. So, like, we did the calculation, we are able to do it, like, 1/5 or 1/10 of the boiler size that otherwise were required. And OPEX, as I mentioned, electricity chill water requirement is less number of sheets, reduces cost of daily monitoring. That's a big cost. Because the larger the area, larger number of plate exposure, we just gotta keep churning on those results, and obviously maintaining that. If you have any deviation in the monitoring aspects of it, that again is a batch lost. So that's again a big plus. Raw material, as I said, cost of material, approximately the same. Cost of resin will decrease drastically. So consumables, long batch durations, no hold times, means reduction of number of bags also. So number of bags we are going to use, for single use, that's going to be low. So that's again a good for consumables. And what are the issues? Obviously, it's not going to be all happy story and good news. It's a dynamic process. That is a understatement, to be honest. It's such a dynamic process that it's very difficult to run a process for 30 days without any glitches. But we are working through it. So it requires thorough process knowledge. So we have to know how the cell is going to function on day 1 and day 30. What is its what kind of impurities does it generate? Does it generate difference? And continuous monitoring. So here the thing is as I said, PAT would be critical. We would need equipments to measure pretty much all the parameters continuously. So that will be very, very interesting. And automation, it's not going to be only necessary, it's actually essential. It will be the thing that will decide if this thing is going to work or not. So we'll be investing a lot of time and energy in automating the whole thing, making sure the bioreactor talks with the home column talks with the TFO filter, all that stuff. So that has to be there. Then the next slide, the connecting upstream, and so one of the issues, again, connecting upstream and downstream, that is going to be a huge challenge, because the bioreactor could be contaminated, the chrome lines could contaminate from the flow from bioreactor, the flow rates we have to be rechecked, then the troubleshooting aspects of it, what exactly, how would we actually kind of troubleshoot if if the downstream process is not working properly and the upstream is actually attached to it. So we have to come up with those kind of technologies. Viral inactivation, no off-the-shelf solution available as of now. It disrupts the continuous flow again, so is there a continuous way to get the viral inactivation? Nanofiltration, again, continuous filtration is going to be challenging. It depends, there's drying of the filter, continuous, how much load can nanofilter take, all that stuff. So the issues are there are many issues. Obviously, we were able to circumvent a lot of them. But obviously it still exists, the scale increases, the issues expands a little bit more. So these are things which we feel it's not a showstopper, but certainly challenging. A lot of challenging, interesting projects. So that's my I think I'm probably right on track on time. So thank you so much for the for your attention. And if you have any questions, I see that 2 questions are there. We'll try to get that by the end of the presentation. If you have more questions, please let me know. Okay, thank you so much. Thanks, Ursula. Up to you.
Thank you, Samir. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the questions tab located at the right-hand side of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second presenter, Lotta Molander, Global Product Manager at GE Healthcare. Lotta Molander is the Global Product Manager covering GE Healthcare's continuous chromatography portfolio. With a background in chemical engineering, she joined GEHC Life Sciences in 2006, spending the first 10 years as section manager and department director within R&D, supporting developmental projects for Exichrome columns and Acta lab scale systems before joining the product management team in 2015. Welcome, Lotta.
Thank you, Ursula. So let's skip to the next one. So my talk today will cover continuous bioprocessing then as one approach to achieve improved process efficiency. And I'll talk a little bit about continuous chromatography as well as perfusion, and I also want to share some of the current experience that we have seen from our end. But to start with, I just want to talk a little bit about why we call continuous bioprocessing one tool in the process intensification toolbox. And as I'm sure you're aware, there are many different drivers for why you would want to improve your process efficiency. And there are also, of course, many approaches to achieve this. And I'm thinking that depending on your specific bottlenecks or your drivers or your constraints, the best tool or the best option for you would vary a lot. For instance, as in the occasion that Samir described just recently, when you're building a new facility, your drivers and priorities can be very, very different to a company that are working only with existing facilities and equipment. And there are a lot of factors that will play a role here. The volume that you need to process every day, or how stable your products are. And what I really want to come down to is that there is really not one solution that will fit all. So you would have to look into your own drivers and find a solution that would fit your priorities the best. So, what I wanted to show you here, this is just a schematic picture of the mab process with some of the different approaches that you can use for process efficiency. You could work with automated or in-light media and buffer preparation, of course increase the level of automation and system control, or any combination of this. And of course, the one option and the one that we will talk about today is the continuous bioprocessing. And we will start with talking a little bit about the continuous capture. So back again to what your priorities are. How can you then determine if a continuous capture step is the right choice for your process? And if you look at the spidergram here in this picture, this is a schematic comparison between what would be a more traditional batch step that you can see with the orange line, a continuous capture step that is kind of straight converted from the batch step, which is represented by the blue line, and also what we call an optimized continuous capture step, where the non-loading steps have actually been shortened, and that is the blue dotted line. And just by looking at this kind of schematic picture, it's kind of obvious that there is no one setup that will provide you with benefits to all your different process parameters at the same time. And that would be the same regardless of whether you're optimizing your step in a batch or in a continuous mode. So what you need to do again here is identify your key priorities before choosing a solution. So for instance, you could easily choose to decrease the amount of resin needed, like Samir was mentioning as well, by something like 50% for the capture step. But this would come at the cost of significantly prolonging your process time. If you want to do it the other way around, you could, of course, shorten your process time and improve productivity that way by at least 30% or more. But this setup will then give you limited gains, again, then in the decrease of chromatography resin needed. But for all the cases when you decide then that the continuous chromatography step is the best choice for you, if you're working with us, we have the solutions from the PD scale here and all the way to up to manufacturing scale. And what you can see in the picture here are the different designs and sizes that are currently available. And on the bottom line there, you can also see the corresponding bioreactor harvest volumes that would fit perfectly with those systems. And this is a slide where I just wanted to show you an example what we're doing with scalability within systems when you're talking about continuous chromatography, which could be super beneficial to use the same or similar setup when moving from like a clinical manufacturing to a commercial manufacturing site. So if we assume that you would like to process one bioreactor volume every 24 hours, and in this case that you're working with the mab select Prisma resin for the mab capture step, you could within the same system scale up all the way from a 200-liter bioreactor and all the way up to a 4,000-liter bioreactor. And the examples here of the amount of mab that you can purify is of course different and will vary both with titer and both with your yield, of course. The key features then for the bioprocess PCC skid that we see work very well when you want to introduce it into a fully continuous process, and that I wanted to highlight here, is first of all the dynamic control function. And that acts as a PAT tool in line that will actually allow you to handle any variations, both in the feed coming in and also any variations between columns. You also have the real-time trending included here, which will allow you to make sure that this part of your process is running very robustly in the steady state. And what we have assured as well is to look into the mabs select resin family, and in this case it's shown here with the mabs select PCC resin. That the performance with the resins is consistent over time, even when you're working with overloading like you do in a PCC setup. Also, these systems are based on a very established platform, which makes them robust as well. And with this, if you choose the most flexible design for this bioprocess PCC feed, you're actually getting multiple systems in one. Since you could choose to run it as a batch system as well, you could choose to run it in straight-through processing mode or in PCC mode. And then the last thing I wanted to highlight is, of course, the reduced buffer consumption that I think Samir also measured that you can get as an effect of working with continuous chromatography. So that was a quick look at some of the benefits that you can get by introducing a continuous capture step. And I would now like to move over to perfusion cell culture and share some recent exciting results from our end on that. But first, I wanted to come back to one of the questions also discussed by Samir. Why is perfusion even an interesting option? And I think in line with what he was discussing as well, the key driver probably is that traditionally, if you go with a fed batch, that would normally require quite a high capex investment when you're working with looking at new facilities. While a high cell density perfusion process with a high productivity can give you the same capacity as a large fed batch bioreactor, but with a much smaller footprint. And that will then significantly reduce the size of your facility. And these are some recent exciting examples for some work here done within the team internally. And this is perfusion data where we have worked with the small-scale wave bioreactors and in TFS mode. And if you look here at the figure on the left-hand side, here you can see what happens when we've been working with a medium that has not been optimized for perfusion. But what I wanted to share here is that even under this kind of sub-optimized conditions, we can manage to push the cell density to above 200 million viable cells per mil by increasing the perfusion rate all the way up to 10 reactor volumes per day. Of course, then 10 reactor volumes is probably nothing that you would like to scale up to the manufacturing scale. So what we did here, if you look at the right-hand side, is that we also did an experiment where we've been working with an optimized media for perfusion instead. So looking there at the right-hand side, you can see that in this setup, we could reach 130 million cells per mil, working with 1 bioreactor volume per day. And if we're perfusing 2 reactor volumes per day, we reach over 230 million cells. So let's look at what we're trying to achieve here. When we're talking about high-performing or optimized cell culture media, I'm sure you all know this, but the key to a feasible perfusion process is really to secure that you can reproducibly achieve this desired volumetric perfusion rate, and that will require very good medium for perfusion. And the performance of the cell culture media is characterized through what cell-specific perfusion rate, or CSPR, the medium can support. And this is the volume of medium that you will need to add for every cell and every day to make sure that the cells are performing in a good way. And if you have a high CSPR value, that will basically mean that the medium is not adapted to the cell metabolism, while when you're looking at a low CSPR value, this will indicate a high performing medium. And if you look at this graph that's on here, this will just illustrate the impact of CSPR on the volumetric perfusion rate. And for our case, that I will go into on the next slide as well, if we assume that we want to work with 1 reactor volume per day, and that we want to operate the process at 50 million cells per milliliter, which is the dark blue line, that will then show us what kind of demands we will need to have when it comes to CSPR for the medium. And if you look at the graph again, you can see that this would then indicate that the CSPR would need to be as low as 20 picoliter per cell and day if we want to achieve these targets. And also, if you compare, if you look at the CSPR value as high as 100 picoliter per cell and day, then you would need to perfuse as much as 5 reactor volumes through the bioreactor each day if you wanted to achieve the same cell density. And this might be feasible in lab scale, but again, this is probably something that would be quite challenging and unwanted to scale up to manufacturing. But with this as background then, let's look at again at some examples where we have working with this high cell density perfusion in the wave bioreactor again, but working with this the developed high perfusion media. And if you look at the left figure here, you can see that the CSPR of this optimized medium is as low as 10 picoliter per cell and day. And that is even lower than what we identified then on the last slide that would be needed for us to work in an efficient way. And if you look over at the right-hand side, you can see that when we are using this optimized medium, we get some really good results for cell-specific productivity and the titers from this setup. So if we're working with 1 reactor volume per day, you can see from the slide or from the picture that the titer is 1.4 grams per liter. And if we go up to 2 reactor volumes, you get 3.4 grams per liter. And the volumetric productivity is as high as 6 grams per liter a day in this specific setup. So these results are quite exciting, I think, and very interesting to go forward with. So that was some details around perfusion as well, but now I wanted to share some current experience with you. And maybe you're aware, but GE has been active in this area, working with continuous bioprocessing for quite some time. I think, for instance, we delivered our first large-scale PCT system already at the end of 2014. So this is to share some of the experience that we have, and especially in the large scale. And this is a summary of the existing bioprocess PCT setups that you can see today with the industry. And what the companies that we're collaborating with have been able to show so far at large scale is that it is possible to scale up from the extra PCT to a bioprocess PCT with maintained product quality and yield. We've also shown that the PCT skids are able to handle any variation in feed and also any unplanned disruptions during these extended runs where they are used. You can also clean them, which is of course super important in an area like this, and you can do it either with steam, if you have chosen to go with one of the larger scale systems, or with chemicals for the smaller scale ones that are not really fit for steaming. And we can also see that continuous capture steps are installed either as a standalone unit, but more commonly I would say we see them implemented connected with either one or more unit operations additional to the PCT step. I think the most common one we've seen is connecting the perfusion bioreactor through the filter setup and then onto the continuous capture step and then to a virus inactivation step. And the preferred control solution so far, especially when it comes to connected processes, seems to be Delta V, but we are working with unicorn setups as well. And for both these control systems, we've been working with automation solutions that is facilitating the connectivity both to the upstream part, but also to the following downstream part. And what I wanted to mention as well is that these systems are used either in like a 24-hour window if they are connected or used as a standalone unit, or up to 20 or 30 days when they are connected to a perfusion bioreactor. And what we've also learned is that when you're working with connecting unit operations in the process, in the manufacturing scale, this will also have implications on how you work in the process development scale. So to support companies that want to work in the PD scale but using existing equipment, we've developed a guideline that is working with IO box connections to be able to utilize any chromatography skids that you have today or any bioreactors that you have in the lab scale. So in this setup that is described in this guideline, we're starting out with a scale that is actually collecting the output from the bioreactor and the filter setup. Once that scale reaches a predetermined weight or volume, that will trigger a signal through the IO boxes that will kick off the capture method on the extra PPC system in this case. Then you collect the elutions from the capture step, and when you reach the predetermined number of elutions, that will again send a signal through the IO box that will start the virus inactivation step. And we've chosen here to do that as a standalone, using standalone pumps and pH sensors, because as Samir alluded to as well, there are no super easy solutions to do this in a very automated fashion for the PD scale. So we're doing it here like toggling between two batch setups, which will give it a continuous process anyway. And once the predetermined time for the virus inactivation step is reached, that will again trigger another signal, and that will start the polishing method on the extra pure. And what we've very recently done as well is that through a customer collaboration, we've added a USDS step to the end of this process. So we will be updating this guideline very shortly to include that step as well. And if you're working with a setup like this, this will actually allow the sample to automatically flow through your entire process. But without adding any additional overarching software, you will not here get any additional support for feedback or feed-forward control, decision support, or handling of any deviations or escape routes. And to focus on that part of the integration as well, we are also exploring alternative automation solutions here for future development projects. And this is a case study that comes from one of our collaboration partners. So this work is done by Rebecca Kimilowski and her team at Merck in Kenilworth in the US. And this poster was actually presented already in 2015, but I think it's interesting enough, so I wanted to show it to you here as well. And as you can read here from the bullet point, the team here has proven that they can both scale up and also integrate a semi-continuous downstream process, and that process will give comparable quality results and yield, but at the same time improving productivity by twofold to compare to the original batch setup. What you can see in the poster as well is some data for using the dynamic control functionality at the PAT tool. And I've included here as well, and if you can't read the reference, you can reach out to me afterwards. It's a reference to an article that Merck has written together with GE, showcasing a little bit more on the work that has been done with pushing the dynamic control feature as far as possible. So what's shown in that article is that the dynamic control can reliably handle variations from cell culture feed with titers ranging from 3 all the way up to 31 grams per liter, which is quite impressive. We sometimes get questions as well on what happens on the lower end if you go below 3 grams per liter, and that is not included in this specific article, but we have through other customer collaborations shown that we can at least go down as low as 0.5 grams per liter if it is a robust process, but somewhere around there lies the limit for the dynamic control. But that gives quite an extensive interval anyway. And something else that I wanted to mention here is that, of course, there are other ways as well when you're looking at intensifying your downstream process, and that is to evaluate new resin options. And I wanted to highlight here the new mab select Prisma resin. that will give you improved productivity, both in batch, of course, but also in a continuous mode. But more especially, this also has improved alkaline stability features, which can be very useful when you want to run the long extended processing time that will be the case when you're working with continuous processes. And that can then potentially require more extensive cleaning to prevent fire burden, and this resin would allow you to do so. And a brief case study as well, when we've been using the Prisma resin in combination with PCC. This is from a poster that was presented at the ACS conference earlier this year in New Orleans. And what we've shown here is that you can work with the Prisma to scale up all the way from a 1 milliliter high trap column into a 5 centimeter acetone column already within the extra PCC system. And you can do this and getting very comparable quality and yield results. And use again the dynamic control functionality and the trend curve to show that you have your process in a steady state and a robust setting. And then the last case study I just wanted to mention. This comes from the BPI East conference in Boston last year. And this is from one of the beta testers for the mabs Select Prisma prototype as it was at that stage. And this customer was evaluating the Prisma resins and choose to use that in a PCC setup as well as in a batch setup. And here again, I have included a reference. And if you cannot read it, please reach out to me afterwards. So this is a reference to the BPI booklet where you can read more details. But the conclusions also from this initial evaluation is that when you're combining the benefits that you get from a high capacity resin like the Prisma and a continuous chromatography setup, the benefits will be improved even further. So that brings me to the summary, I think. And what I wanted just to say here and agree, I think, with Samir's conclusion as well, is that continuous bioprocessing is definitely one viable tool in this process intensification toolbox. And with the right conditions, this can really provide great benefits from an overall process perspective. Also working with single unit operations, you will get some of these effects and some of the nice benefits, but the true benefits seem to come when you are really connecting multiple steps, working in a complete continuous or a hybrid process, but definitely a connected process. And what we've seen also in these slides are case studies that show that we can significantly improve the productivity through perfusion setup when working with a high capacity medium as well. And you can also see that the PCC technology is implemented and proven from process development to manufacturing scale. And when you go for a continuous chromatography step, that you can get additional benefits with working with high capacity resins as well. And also some insights into the current experience that we see that show that the connecting unit ops work very well, as I said, either as standalone or as an integrated part of your process. And with that, I only wanted to thank our collaboration partners with Merck and of course the team here at GE Healthcare working both with automation, upstream and downstream as well. So thank you for listening, and then I think back to you, Ursula.
Thank you so much, Lotta. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that they can still send their questions via the questions tab on the right-hand side of the screen. The first question is, do you foresee any issues with using continuous bioprocessing in biosimilar products for which the innovator has a batch process? Samir?
Yeah, hi. Great talk, Lotta. For the question, I think that's going to be an interesting thing, especially for all the biosimilar companies. I mean, when we are looking at in the production process that needs to be followed for our production in comparison to the innovators, that will be the first question. Is that how much similarity does the production process have with the innovators? So the thing is, the way we think about it is it's although the production process is continuous, but it's still the basic science is the same. The cell line that is developing that or producing that product is actually a recombinant DNA, like, or the CHO cells, or like, is it going to be like, whichever cell line we pick, like, that's going to produce the same thing, like, and what we need to prove from our end, from the manufacturing end, is that the product that is being produced in Perfusion is the same as in the batch. Or day to day, like, the product variability is not that high. And that's something that, like, at least we have been successful in doing that for the products that we have. But that is something that each company has to do with its own research and figure it out. Then the next part is the continuous downstream part of it. Now, continuous downstream, like, actually, as I think, like, in our talks we discussed, it's a lot of, like, it's actually the principles are the same. It's the same protein A column, like, that we are using as a resin. We are trying to capture that. Viral nitrogen step is going to be the same. So it's only like, just the way we do it. I mean, as I said, it's going to be like a lot of micro batches. I mean, like, we burn and US FDA has in the future been acceptable to, like, smaller lots of DSP and then being pulled together. So, I mean, this is pretty much the same case here also. So from just the production process itself, I think continuous should be fine. And we have actually, as Enzene, like, we did approach US FDA for their guidance on this too. So, I mean, like, it has been pretty positive. So from our end, I think, like, we are fine with, like, having a continuous process. For biosimilar, that the innovator might have a batch process, and then also, like, there's, like, the biosimilar industry has to make some changes to the process to make it cost effective. Otherwise, like, what is the point of actually having how can we actually make the product as expensive as the innovator company and still sell it at a cheaper rate? So that's again a business case for that.
Thank you, Samir. What trends do you see developing for continuous processing? Lotta?
I think what we see is that there is a move from starting out with evaluating more continuous unit operations and to see how they would fit into your process. But I think recently we are talking and focusing more and more about connecting and integrating a complete process. And any kind of discussions connected to that, I think, both the implications for the process development that we were mentioning before, what kind of control strategies need to be in place to do this, because you will get a complex process, efficient but complex as well. Automation solutions is something that we discuss a lot, I think. Also sensors and analytics needed for this. And again, as Samir mentioned in his talk as well, we are talking a lot about process economy right now, and where I think that the companies we are talking to and working with are getting more and more experienced as well, and a bit more critical maybe when evaluating these as possible solutions for their process to really choose the solution that fits with their specific needs. So to summarize, maybe I think more, yes, sorry.
Thank you. How would you define a batch in continuous process? Samir?
I think that's a very interesting question. I mean, like, I think it's been the question that has been asked in probably all the forums that I've given, like, this kind of a talk. And I think people, like, it's a justified question, to be honest, like, what exactly, how would you define a batch? Because you're actually getting the product out, like, every second. So do you pull the entire lot and then, like, call it a batch, or what exactly with the batch? I mean, the thing is, it's again, like, it has to be done through the science, like, that we are developing, the process that we are developing as manufacturers. I mean, we have to give FDA or the regulators the justification that, like, whatever we are collecting and calling it a batch is consistent. I mean, the regulators require a uniform, product quality in the batch. I mean, that's what they require, and that's again what we need to prove from our end. So it's going to be a lot of experiments that needs to be done, taking different parts of the continuous process and see where the product quality is, or collecting samples on a daily basis or even hourly basis to see what is the different variations in the product quality or things like that, and then kind of have enough data to make it a convincing presentation to the regulators. So I think the batch in continuous is going to be a little challenging, but I don't think it's going to be a showstopper again.
Thank you, Samir. Do you see continuous chromatography being implemented in other areas than MAB capture? Lotta?
Yes, I think the primary interest is still around mab capture or affinity capture at least. But for the last 1 to maybe 2 years, we have seen an increasing interest in other areas as well. Of course, things like ion exchange that could come as a second polishing step in the mab process, but also an increasing interest, I think, from vaccine processes, where we have done some work with collaboration partners looking at virus purification with the capture core, for instance, and working more than in the flow-through mode than the bind-dilute mode that we'd normally use for the affinity capture, of course. So it is changing, but the primary focus is still, I think, affinity capture, but moving into other areas as well, at least for evaluation and investigating.
Thank you. For high-volume, low-cost products, does continuous production help for the site?
I can take that. So yeah, I think basically even if it's high volume, it's actually a pretty good process. So although the scale of the bioreactor might be small, the high volume does not prevent it from making more of that. And also, the thing is, it depends on the productivity of the process. If it's a high producing cell line, we can actually have very high volume of it. And as I said, the plant that we are making, that is going to produce 450 something kilos of mabs, and that's that's a pretty high volume product that we're looking at. And you should see that it's only around 10,000 square feet of actual production space, not even less than that. So that's the whole beauty of it. So we can actually scale up a little bit, scale out. That's a very good possibility. We can actually make it. And the whole reason we kind of chose it for biosimilars is because of the low cost part of it. And again, as the biosimilar industry grows, it will be the cost pressure would increase. So we will need to manufacture at a very lower cost and sell it at a much lower cost than the innovator for sure.
Thank you, Samir. So you have no tank between upstream and downstream.
So yeah, I guess I'll take that too. So right now, I want to say yes. There's no tank, but unfortunately, we are at that process, where we actually have a small tank. I mean, just so that the circuit breaker, something happens, we can, we have some time to react, right? I mean, so that is the whole idea of that circuit breaker source tank. But again, it's not a huge tank, it's not going to collect the entire days of culture, 3 days of culture. What we'll have is a very small tank, what we have, and then the thing is that is actually used for just a circuit breaker. But the thing is, going forward, at least in the production phase, once we have enough data, on the process, then we will kind of eliminate all the tanks, because then basically even if we have a problem, there should be enough automation in the process to actually divert the flow or make some changes to the process to kind of accumulate that. So that's the goal of it.
Thank you.
Maybe I can comment on that one as well, Ursula. I think what Samir is describing is very similar to what we're seeing with a lot of companies as well, that there is a small break tank for sort of security reasons, or making sure that you can adapt to any changes in flow, for instance, and handle small variations. But in accordance with what Samir is describing here, we also see that there is a strive to go away from the big whole tanks and go into smaller break tanks instead, and trying to automate and scale the process so that flow will fit the entire start to finish flow or end-to-end process.
Thank you. The next question is, thank you for the talk. Can you tell me more about what kind of BAT tools used?
Yeah, I can start again, Lotta, if you want to join that, I think you had a slide on the PAT tools that we have. So right now, I think what we are using is pretty basic, cell count, all the metabolite concentration that we have for the bioreactors. We are looking at different probes. One of my favorite one is a Raman Probe, that actually, it's not exactly a direct measurement, but actually it could be an indirect, once you get the knowledge, once we calibrate that probe, it could give us a lot of online information. So that is one kind of a PAT tool. Also, a lot of automated samplers. So basically, because eventually the process that we're going to do for 30 days, it has to be monitored on an hourly basis. So I mean, having people there all the time might be risky for the process, and also it's kind of introduces human error. So the automation would be. So we will looking at automated samplers for now. So those are the two PAT tools that we are looking at. We are looking at I mean, whenever a new technology comes in, we're more than interested in looking at that.
Thank you, Samir. Before I finish the webinar, I would like to ask our presenters, Samir Varma and Lotta Molander, if they have any closing remarks. Samir?
Yeah, so my closing remark would be I mean, I just want to make sure that people do understand that this is actually going to be really interesting. I mean, this whole continuous bioprocessing, as I said before, this could be the disruptive kind of technology that the bioprocessing industry is looking for. And as more and more we think about it, more and we actually get into it, I think a lot of interest around the world is there, and I just feel it's that this is going to be the future, and the future looks really interesting and really different, to be honest. I mean, we will be looking at plants that are much smaller in size, small local plant that is actually manufacturing for the local needs. So those kind of things, that's what the continuous bioprocessing will look at it. But again, it's a very challenging process, and obviously, it's going to take some time to get all the things worked out.
Thank you, Samir. Lotta, do you have any closing remarks?
Yes, I think maybe I just to mention that I think this, to agree with Samir, this is a really exciting area, and to be working in this area where we are talking about connecting processes and going to find an end-to-end process is super exciting. And the demands that this will put on automation and all of the data handling and everything around this is a very interesting area, and I think that also fits very well with the current focus of the overall GE business. So it's a perfect match for us, and it's a very interesting area for us to be in. And the ongoing customer collaborations that we have are really exciting stuff. So there are some really nice developments going on here. So I agree with Samir. It's an exciting area to be working in.
Thank you, Lotta. I would like to take this opportunity to thank our presenters, Samir Varma and Lotta Molander, for sharing their knowledge with us. I would like to remind our audience that they can view this webinar on demand by visiting biopharma-asia.com. Thank you, everyone. Have a great day.